Hello everyone, welcome back to the BioTuber. I am Swastik and today we are going to continue with our topic nucleic acids and nucleotides. So in the last class we talked about a general introduction to the nucleic acids and we touched upon nucleotides also. In the nucleotides we talked about the three components, that is the nitrogenous bases, sugars and the phosphates, and we studied in detail about the first component, that is the nitrogenous bases. In today's class, we are going to focus on the second component of nucleotide, that is the sugars, and we will particularly focus on the sugars that are specific to the nucleotides and not the other sugar molecules. So in case of nucleotides, there are two types of pentose sugar that are seen, and from pentose sugar we can know that these are 5 carbon sugar molecules, and the sugars are in general sweet tasting compounds. So let's focus on the pentose sugar. In case of nucleotides, there are two types of pentose sugar, that is the ribose sugar and the deoxyribose sugars. The ribose sugar in case of RNA, whereas the deoxyribose are found in case of DNA. And there is not much of difference between the ribose and deoxyribose. The only difference lies in the absence of a hydroxyl group in case of DNA at the second position of the ring or second position of the carbon. Now we will look into the details of the differences in this class only, and we will talk about them in the later sessions. Now, all the naturally occurring forms of the RNA or DNA or nucleotides are D isomer, and to be more specific, these are beta D ribose sugars or beta D deoxyribose sugars. So what is this classification, the beta D? So the D classification is the same as the DL classification, and it is actually seen if we look the sugar molecules from the linear point of view. If we look into the linear structure of the pentose sugar, then the D or the difference of the D and L form can be seen. So this difference is again in comparison to the D or L form of the glyceraldehyde. As we have studied in our earlier class regarding the amino acids D and L form, the same concept is actually followed with the ribose sugar also. So in case of this ribose sugars, this group, the terminal group as I've highlighted, if this is similar to that of the glyceraldehyde, based on that we will give the D or L form. Here we can see the OH group or the functional group OH group is present on the right, which is similar to that of the D form of the glyceraldehyde, and thus this linear structure will be called as the D isomer. Whereas if we look into this particular structure, the OH group is on the left, which is similar or resembles the L form of the glyceraldehyde, and thus it is called as the L isomer. So there is more preference towards the D isomer in case of DNA and RNA. So I have now the ribose sugars or saccharides in general can remain in both linear form or cyclic form, and it has been also noticed that the cyclic form is preferred more over the linear structure because of the presence of the functional groups, and because of the preference, the more stability is seen in case of the cyclic compounds, the cyclic form is preferred. Now this cyclization actually takes place by the formation of either a hemiacetal or a hemiketal. The hemiacetal is formed if there is a aldehyde group, whereas a hemiketal is formed if there is a ketone group. So if this aldehydic group that is seen on the terminal is linked with any of the functional OH group present in the chain, it forms a ring structure, and thus the hemiacetal or hemiketal formation is there. In case of particularly ribose, the hemiacetal structure is formed when this aldehyde group forms a link with the C4 carbon, that is the fourth carbon in the chain, and thus it resembles the furan structure, and thus many a time the ribose sugars are called as furanose molecules. If this cyclization or this hemiacetal formation would have taken place with the fifth carbon, then it resembles the pyranose ring. So in case of ribose, we will study about the furan ring structure. Now since the D form is preferred in case of the ribose, I have demonstrated the D form of the D cyclic form of the ribose sugar here. Now this beta, so the alpha beta nomenclature basically depends on the presence of or how the OH group is actually present around the first carbon. So in case of this is the alpha form and this is the beta form of the cyclic D-ribose. And you can notice that in case of alpha, the OH group is directed downwards, whereas in case of beta, the OH group is directed upwards. So if these are planar structure. So if this is the plane of the ribose sugar, then if the OH group is directed upwards at the first carbon, then we call it as the beta structure. Whereas if the OH group is directed downwards, then we call it as the alpha structure. So this alpha beta difference is also called as the epimers, which is a part of the stereoisomerism, and by definition epimers are those isomers which differ specifically at one particular position of the carbon in case of hemiacetal and hemiketals of the saccharides. So the alpha beta differences is because of the presence of the hydroxyl group. Now it was not until 1909 that it was found out that the D isomer is actually seen in case of the all naturally occurring nucleic acid or ribose sugar, because when it was first synthesized by Emil Fischer and his colleague, they found or synthesized the L form, and it was not until 1909 that it was found by Levine and Jacob that the D isomer is actually seen in case of the naturally occurring ribose sugars that are seen in case of nucleotides. So this is the nomenclature or which is preferred, that is the beta D ribose sugars are preferred. And now we will look into actually the DNA and RNA. What is the difference between the DNA and RNA? So I will remove this and I will make them both the beta form so that it will be easy to understand, because whether it be DNA or RNA, only the OH like the beta D ribose or beta D deoxyribose are seen. So if this is directed upward, that is the OH group is directed here. Now these are both the beta D ribose sugars. So now this is the structure of the pentose sugar that is seen in case of the RNA, where each carbon atom is occupied by a hydroxyl group. So this is the structure of the pentose sugar that is seen in case of RNA, that is the normal ribose, that is beta D ribose. But in case of DNA, the second position actually lacks a hydroxyl group and there is presence of a hydrogen. Rest is the same as that of the ribose or the RNA, but there is only difference lies in the OH group at the second position. This may not look significant as such, because what is there in like replacement of one OH group with a hydrogen atom? But this is very significant if we look into the stability point of view. Because of this replacement of a hydroxyl group with a hydrogen, the DNA is preferred as the genetic material and not RNA. RNA no doubt is formed during the process of transcription, but the stability of DNA is multifold increased by the replacement of the hydroxyl group with a hydrogen atom. And thus this is now the DNA, that is deoxyribonucleic acid, that is the absence of oxygen at the second position of the carbon. So okay, so this is the structure of the pentose sugar that is present in case of the DNA, that is it is 2-deoxyribose, that is the second position of the pentose ring lacks a hydroxyl group and the hydroxyl group is being replaced by a hydrogen atom in the pentose structure or the ring structure. Now again there is one important thing that we must notice, that the naming actually of the carbon atoms in the ribose sugar is not the same as that of the nitrogenous bases. In case of ribose sugar, the carbon atoms are named as C1-, C2-, C3-, C4- and C5-. And this is also called as C1', C2', C3' and C4' respectively, because we need to differentiate it from the carbon atoms that are seen in case of the nitrogenous bases. In the nitrogenous bases also carbon atoms are present and they are named as C1, C2, C3 and so on. So to distinguish them from the to distinguish the carbon atoms present in the ribose sugar from that of the nitrogenous bases, we give the prime symbol or the dash we generally call. So it is a simple thing, but it is important because when we are referring to particular position of the carbon, we can differentiate with the help of this prime from the nitrogenous basis carbon. So this is all about the the beta D ribose sugar and the beta D deoxyribose sugar, and how DNA and RNA are different in presence of the hydroxyl group. One more thing that we will study about the ribose sugars is the sugar puckering. So let's have a look at the sugar puckering. Okay, so let's discuss about the sugar puckering. So what is basically sugar puckering? Pucker means if there is presence of any kind of wrinkle, ridge or twisting, that is called as puckering of any structure. In case of sugars, or in this particular case the ribose, the puckering is because of the presence of the functional groups. Because when the functional group are present in the planar structure, there is more of a torsional strain that is experienced by the structure, as a result of which that is less stable. So the puckering basically enables a more stable structure for the ribose sugar. Earlier I have mentioned that ribose is kind of planar, but it is not completely planar. It is, all the atoms are not planar to each other. Some of the atoms are planar while some are not in the same plane. There is some kind of puckering, and that is because of the presence of the functional groups. Now, if we look into the ribose or deoxyribose, the puckering is basically classified into two different types. The first one is the envelope or twisted type, and the second one is the endo and exo type. So in case of envelope puckering, we see that 4 carbon atoms are coplanar to each other, whereas the 5th carbon is away from the other 4. So if we look at the structure of the puckering, one of the carbon atom will not be in the same plane, whereas in case of twisted, 3 carbons are coplanar, whereas 2 others are away from the plane. So if I try to demonstrate using some of the papers that I have taken, so if this is the structure of the ribose, where the O, this is the first O group, this is the first carbon, second carbon, third carbon, the fourth carbon, and if I fold it along the line that I have drawn, so this is kind of an envelope now. As you can see, this resembles the structure of an envelope, and thus the name the envelope. And you can see that the 4 carbons, 1, 2, 3, and the fourth one which is attached to the last carbon which is attached to the C4 dash carbon or C4 prime carbon, they are in kind of a plane, whereas this particular carbon, the C3, is slightly upwards or away from the plane. So this is the envelope configuration. Now similarly, if I look into that, if I look into this particular ribose sugar, then this particular twisting, there is kind of twisting that is seen in case of this molecule. So these two are in one, these three, that is the first carbon, the fourth carbon, and the fifth one which is attached to the fourth carbon is kind of in one plane, whereas the C2 and C4 are in different plane. There is some kind of twisting that is seen in case of the C2 and C3, as a result of which this is the twisted form. Now, so let's look into the endo and exo classification then. In case of endo classification, the C3 and C4 that I have twisted, or those 2 carbons or the 1 carbon that is away from the plane, if they are directed towards the C3-C4 bond, C4-C5 bond, then it is called as the endo classification. Whereas if the one which are not in the plane are directed away from the C4-C5 bond, they are called as the exo puckering. Now, if this is the structure, this is the C4 and C5 bond that I am talking about. So if the direction is towards this particular bond, then it is the endo classification. Whereas if the twisting or the envelope is away from this C4 C5 bond, then it will be considered as the exo classification or the exo puckering. So in case of purines, there is more preference towards the C2 endo, C2 endo, that is the second carbon. This carbon will be directed towards the C4-C5 bond, whereas in case of pyrimidines, the sugar has, or the ribose prefers the C3 endo puckering. In case of RNA, if we look, the RNA mostly prefers, or the predominant form on RNAs are the 3' endo, and this is how actually the puckering is seen. So now that we have looked into the sugar pucker or sugar puckering of the ribose sugar that are present in the nucleic acids, we will further study how actually these sugar molecules are linked or bonded to the nitrogenous bases and the phosphate groups in the coming classes. And this will be all for today's class. If you have any query, you can put your query in the comment section and I will try to answer them. If you like this video, subscribe to my channel and share with others. Thank you.